Hey guys, so we're continuing our look at genetic diversity, and in the next two videos, we're going to be looking at Mendelian genetics. So when we're talking about Mendelian genetics, this really connects a lot with our evolution unit and even our next heredity biotech unit. So we've got this concept of common ancestry. So when we're talking about common ancestry, we start with the basis of DNA and RNA carrying the genetic code. And it doesn't matter what type of living organism we're talking about, we still got this base molecule that everything is designed and written from. And so it is shared by all living systems, this genetic code, the DNA, RNA concept. Gregor Mendel studied inheritance, and he created two key laws that are actually applied to the study of genetics. Gregor Mendel is, of course, considered the father of genetics because he was the first one to really put a basis to all of this. People always knew that somehow or another information, some characteristic, something was transferred in creating new living organisms, but there was really no understanding of how that worked. And Mendel came up with this. Of course, it got hidden for a little while because Mendel was doing his work about the same time Darwin was doing his and came out with his groundbreaking concepts at the time. And so Mendel's kind of got pushed aside and then was rediscovered a little later. But nonetheless, Gregor Mendel is the one who did these studies and came up with these concepts. And so we've got two key laws that he came up with. So a little bit about Mendel. He was an Austrian monk, and he discovered the basic principles of heredity. Now, the reality is we were really lucky that he worked with what he worked with, because he might not have been able to figure it out if he'd used something different. So he experimented on pea plants. Why? Well, first of all, do you really think that a monk, therefore part of the church was going to encourage him to intentionally breed individuals, organisms, things like that, just to see what happens if you mix this with this? But he experimented with peas because there were lots of different varieties. He could control the mating. There wasn't going to be any oopses. Somebody got into somebody else, right? And they had a relevantly short generation time. Think about it. Just for humans, when they reproduce, you've got a 9-month waiting period before you can start seeing any outcome. And then they still have to grow before you see a lot of the characteristics. So humans definitely would not have been a good choice. Peas work pretty quickly. So when we're talking about these peas, he tracked characters that only had two options. So he made some selection. He picked things that it's like it's going to be this or this, this or this. So for example, the color, were they white or were they purple flowers? The shape of the seeds, were they round or wrinkled? To help control his experiments, he used what were called true breeding plants. And a true breeding plant means if you cross two of these true breeding plants with each other, you're going to end up with only those things. We're saying that they're all purple, they're all round, they're all wrinkled, they're all white. There's no option of getting something else from it. So true breeding organisms produce offspring of the same variety over many generations of self-pollination. So true breeding purple pea plants will only ever produce purple offspring if they self-pollinate. And that brings us to the idea of generations. And this is really important to remember as we work with genetics problems. So the P generation would be those true breeding parental generation. So the purples and the whites, the rounds and the wrinkleds, right? The F1 generation, known as the first filial, these are basically the first set of babies, the children of the parents. And then the F2 generation, second filial generation, would be if we took two of these children, these pea plants, and we cross them with each other, then we end up with our grandchildren, our second filial generation, our F2 generation. Okay. So parents, true breeding, F1 would be the children, F2 would be grandchildren from the children. And then we had a scientist who came in, and his last name was Punnett. That's where the name comes from. So he came up with this idea to help with the probability of these genetic crosses. So this is not something Mendel came up with. This is something another scientist came up with based on things he'd learned about genetics. And he said, hey, this seems to work. So Punnett squares are diagrams that are used to predict the allele combinations of offspring from a cross with known genetic compositions. So few things to remember when we're talking about this. Capital letters denote dominant traits, lowercase letters denote recessive traits. That's very important to remember when it comes to genetics problems. Here's a few key vocabulary terms you've got to know, got to understand, got to keep clear. We're going to start with homozygous. Homozygous is an organism that has a pair of identical alleles for a character. For example, we could have homozygous dominant, big A, big A. We could have homozygous recessive, little a, little a. Heterozygous, an organism that has two different alleles for a gene, and that would be, for example, big A, little a. Then we have the term genotype. Genotype refers to the genetic makeup or the alleles, the letters of an organism. So what do the actual letters say? Do they say big A, big A? Do they say big A, little a? Do they say little a, little a? What do the letters physically say? Phenotype is the physical appearance, phenophysical, an organism's appearance, which is determined by the genotype. Are they purple flowers? Are they green horns? Are they wingless? It depends on what you're looking at, obviously, but what do these traits physically look like? And so then you would perform a test cross if you're trying to figure out what some sort of undetermined organism's genotype is. So for example, here, we know that this is yellow. It has a capital Y, but we don't know if it's capital, capital, or capital lowercase. So if we perform a test cross, we do it with the homozygous of the opposite. So here we have homozygous recessive green. When we cross them, we look at what would we actually get. So if it was capital, capital, when we did a test cross, we would get all yellow. Whereas if it's capital lowercase, we would get part yellow, part green. Okay. So when we come up with the principles of heredity, Mendel he developed two fundamental principles. The first is the law of segregation. The second, the law of independent assortment. Okay, so those are his two key laws. And we're going to discuss those as we kind of go through what he did so you understand them. So we've got this same diagram. Remember, this is that diagram we showed with the test cross of the P, F1, and F2 generations. So he noticed that the cross between purple and white true breeding plants produced only purple F1 offspring. So the question then arose, did the white character disappear? What happened to the white? And the answer is no, the white did not disappear, because if he allowed two of the purple F1 generation to self-breed, self-pollinate, then we ended up with white and purple in the F2 generation. So that white characteristic came back in the next generation. Well, how could that happen? So that's where the idea of dominant versus recessive kind of came from. He hypothesized that the purple flower must be dominant trait to the white flower, which then would be a recessive trait. So when we're talking about dominant trait, we're talking about a trait that masks or covers the other trait. That means a recessive trait can only be seen in the homozygous recessive condition. So he performed the same crosses for each of the 7 characteristics of the pea plants that he used. And here you can see a listing of with illustrations for the 7 characteristics. And he found that that happened over and over and over. It wasn't just purple versus white flowers. It happened with each and every one of these traits. He also found that the F2 generation was always a 3-to-1 ratio, where you got 3 of the dominant trait versus 1 of the recessive trait. So to explain the 3-to-1 ratio, he really observed that there obviously are alternative versions of genes or alleles that account for that variation. So we've got to have at least 2 different forms. For each character, an organism inherits 2 copies. So they have to get 2 alleles of a gene, and 1 allele comes from each parent. If 2 alleles at a locus differ, then the dominant allele determines the appearance and the recessive allele has no noticeable effect. So we're saying that the dominant one covers up the recessive one. This led to the law of segregation. The 2 alleles for the same trait separate during gamete formation and end up in different gametes. So we're making sure that one from mom, one from dad, well, when we're actually making eggs and sperm, we start separating those things out. And remember, that's how we take it to that haploid concept that we talked about with meiosis, is we are separating them, so they're going to end up in different gametes. So a little closer look at alleles. And so here you see we have 2 chromosomes, 1 from each parent, and they have certain genes or alleles on them. So somatic cells are diploid, 2 copies, right? 2N. So they have 2 copies of the chromosomes. Alleles are the different versions of the gene. So is it tall or is it wrinkled versus smooth? The 2 different versions. Sometimes the versions may be the same, like the tall. So according to the law of segregation, true breeding plants have 2 identical alleles. So capital P or lowercase. Each gamete for the generation contains 1 allele for the flower color. Now, obviously, like in these true breeding, they're both the same allele. They're both capital P, but there's still just 1 copy in the gamete. The F1 generation then became hybrids. Hybrid, same as saying heterozygous hybrids. We have a capital and a lowercase coming together to make our F1 generation. And then the F2 generation, if we take two of these purple flowered plants from the F1 and we cross them, we end up with our 3-to-1 ratio. Okay, that's it for this session. Hopefully that helps you understand this so far, and I'll see you in the next Mendelian genetics lesson. Bye.